Good evening, everyone. Emory Moynagh here for In His Image, and welcome back to Beyond the Basics, a creation podcast. I promised that we would be doing more science content, and here it is. Today we're going to be reading a paper by Gordon Wilson of New St. Andrews College in Moscow, Idaho, and the phrase Moscow, Idaho may have sent up some alarm bells for some of you. Yes, he does teach at Doug Wilson's college. Yes, he's Doug Wilson's brother. But I've interacted with Dr. Wilson, and he is a very measured, thoughtful gentleman. I've had no issues with him, and even realistically, I think the attacks on his brother are overdone. Let's put it this way. I don't always agree with Doug Wilson. Matter of fact, I don't always appreciate his choice of words, and I don't always appreciate his way of putting things and saying things, and I don't agree with everything he says because I'm not Presbyterian. But the fact that everyone on the left in the church hates him tells me that he's doing something right. So I will at least pay attention to him, because the enemy of my enemy is probably my friend, or at least a potential friend. So I'll at least pay attention to what he says. But Gordon Wilson is I think he's professor of biology out there. He also has the popular film series, The Riot and the Dance. Gordon Wilson is the one behind that. So if you are into The Riot and the Dance, Gordon Wilson is the guy who's been behind that, so you may already be familiar with him. One of these days I am going to get The Riot and the Dance and actually review it. I haven't had an opportunity to do so as yet. But this paper is fairly short for a science paper. It's only like 4 or 5 pages. We will not read the whole thing. We're just going to read some excerpts from it and talk about the excerpts from the paper. The title of the paper is Genome Equivalence and Speciation, Proposing a New Criterion for Baraminology. Now, I've talked to Wilson several times, and when I've talked to Wilson, and maybe I'll get him on the program one of these days. That'd be amazing if I could. He's always expressed some general concern at how baraminology is proceeding, which, given that I have some of those same concerns, we get them all for that reason. But the concern he has expressed is that we are setting our baramins are too wide. In other words, we're including organisms that probably aren't in the same baramin, and he wants to fix that. In order to do that, he is going to talk about what are called taxonomically restricted genes. We'll get into that in just a minute. So let's have a look at what Dr. Wilson has to say. I'm going to read the entire abstract, and then we'll just read snippets from the paper. This paper explores the inability of microevolutionary mechanisms to generate novel genetic information. This has practical ramifications on how baramins are delineated. In light of the limits of macroevolutionary mechanisms, it is argued that current methods in baraminological research overlook this fact to some degree, when holobaramins are often concluded to be at the taxonomic level of family. These results appear to ignore, in some cases, significant disparities in the genetics of the family's members. Because of these inconsistencies, a new criterion in delimiting boundaries of baramins is proposed, genetic equivalence, and is to be used in conjunction with the existing criterion synapomorphy. The guiding principle is that natural processes do not generate new genetic information de novo. The absence within a group of taxonomically restricted genes in a similar species would determine that it does not belong to the group under consideration. This would be a powerful indicator in eliminating species that seem to fit the general morphology. Comparative morphology is a great starting point, but because God created all diversity in nested hierarchies, it is difficult to determine at what level similarity is due to common design and what similarity is due to common descent. It is argued that genomic equivalence can determine the boundaries of a baramin that would include all the genetic variation that is empirically possible within a shared gene pool. This is not a proposal to return to a fixity of species concept. Equivalent genomes can produce a wide array of phenotypes due to allelic differences, differential gene expression, genetic recombination, etc., analogous to how one zygote can produce hundreds of cell types. This would require much work, but should be more fruitful in delimiting created kinds. Now, let me just break down what he's arguing here in a very, very basic way. He's arguing for the use of genetics in baraminology, which is not being done right now very much. It is a little bit. Dr. Cherhati is doing quite a bit of work with this, but Cherhati's about the only one. And I really respect Cherhati for what he's doing on this. Another person I'd love to get on the program one of these days. But Cherhati is still publishing on this. Wilson is trying to point out that, yes, we need genetics. And the problem is statistical baraminology, as it stands right now, is not incorporating genetic data at all. Now, I know of some things that I can't really talk about, but there are some people trying to figure out ways to incorporate genetics into statistical baraminology. I have my doubts about it, but we're going to go ahead and just I want to reread a section from the abstract because it's important. These results appear to ignore, in some cases, significant disparities in the genetics of all the family's members. In other words, what he's saying is some families have widely disparate genetics, and this is being ignored by statistical baraminology, which is true. That's completely true. Because of these inconsistencies, a new criterion in delimiting the boundaries of the baramin is proposed, genetic equivalence, and is to be used in conjunction with an existing criterion, synapomorphy. And a synapomorphy is a shared derived trait. So it's a shared morphology. So they're looking at shared morphology plus shared genetics. So basically, a synthesis of morphology and genetics. That's what he's going for here. The guiding principle is that naturalistic processes do not generate new genetic information de novo. That's true. That's true. That is absolutely true. So the absence within a group of taxonomically restricted genes in a similar species would determine that it does not belong to the group under consideration. This we'll get into more as we go through the paper. So I'm going to highlight just a few things. I went through this paper and I highlighted it up. And I want to just go over some things about the paper that are interesting and hopefully will be of interest to my audience as well. Right? That's the goal. So here's the first section I want to quote. And this is from the headed section, Implications of Genetic Information and Speciation. The problem lies not in the morphological disparity or in the number of species produced, rather than the problem lies in whether we think adaptive radiation will somehow generate more specified complex information during the diversification process. This crucial point cannot be overlooked in defining baramins, because the Hebrew word min means kind. We have quite a bit of interpretive wiggle room when deciding how many smaller Russian nesting dolls, quote, species, can fit into the bigger Russian doll kind. But regardless of the number of smaller dolls that can fit into the big Russian doll, we know that new genetic information will not arise naturalistically as we unpack the dolls. Bingo. Okay, that's important. That's important. Because some of these kinds that have been delineated are really broad. Now, some of them have been delineated with very little genetic evidence or no genetic evidence and no hybridization evidence. They've just been done with morphologies. This particular, I'm thinking of the angiosperm one, which is like something nuts, like a couple hundred species, which in plants maybe isn't insane, but I think he did grass, a subsection of grass. Todd Wood did a subsection of grass, and it was like 1,000 species or something like that in one kind. That's a bit speciose for me. And this is part of the reason I don't like morphology, because we don't know at what scale we're looking. If we're looking at the right scale, that might break up. If we're looking at the wrong scale, it might not break up. And of course, the character problem, which I'm working with someone on that. There may be a paper coming out at some point on that. But there's all kinds of problems with morphologically based baraminology. I think genetics is a better way to go. Genetic data is not perfect, but I think it's better. And I think Wilson is getting that. I think Wilson understands that, and that's why Wilson is going for some of the things he's going for. The second thing that I wanted to point out from this paper, quote, family-level baramins is not the issue. That is not where the fundamental difference of opinion lies. What I cannot accept is not so much the degree of branching, but rather the significant differences in the amount of complex genetic information between members of the same putative hollow baramin. I do not mean differences in allelic frequency. So what he's saying is, he thinks that there's too much differences between members of the same, quote, hollow baramin. Now, I want to explain something to you. The baramins that you hear most creation scientists talk about, particularly the young Earth evolutionists, Todd Wood, Kurt Wise, Neil Duran, Ken Colson, Matt McLean, and people of that ilk, are completely divorced from Scripture. And by completely divorced, I mean there is no connection whatsoever to Scripture. It's completely divorced. There is nothing there at all. A baramin is a potentiology, is the actualization of a potentiology region. And there's a bigger definition, but I won't go into that right now. We'll talk about that in another video. And a hollow baramin is just, a group of organisms that cluster together based on morphology, essentially. There's no reference to Scripture whatsoever. These groups that they're defining have zero to do with Scripture. Nothing. Get that through your skulls. If you get nothing else out of this video, get that. The baramins that Wood and Wise and McLean and Colson and Duran and everybody else and Paul Garner and whoever all else is in this group talk about have nothing to do with the biblical text. Nothing. Now they're defining them, and they're saying that, yeah, we're doing this because this text says kinds, but the kinds they find have zero to do with the biblical text because the definitions they use have zero to do with the biblical text. Nothing. Okay? Understand that. So when he says same putative holobaromen, he's talking about a group that is completely divorced from the Bible, which is part of my problem with the whole statistical baraminology method, is it is so far removed from Scripture that I don't trust it. Particularly since it finds that apes and humans are the same kind regularly, which Todd Wood just did a study on that, which we are going to talk about at some point. I don't know when exactly we'll get to it, but we will be talking about it at some point. Moving on here. So here's what he proposes. Here's what Wilson proposes. If species 10 also has genes Y and Z, with neither gene having a homologue in species 1 to 9, then Y and Z are taxonomically restricted genes. So basically what he's saying is in a group of 10 species, if 9 of them do not have genes Y and Z, but number 10 does, those two genes, Y and Z, would be considered taxonomically restricted. In other words, in this group, only one species in that group has those genes, so it's restricted. As such, species 10 should not be included in the same baramin. Evolutionists focus on similarities because they assume continuity. That is why they ultimately concluded one evolutionary tree over 150 years ago. Creationists believe in discontinuities, but they cannot agree on where they occur or how to recognize them. This is key right here. If you get this, you understand why creation baraminology has gone off the rails. The evolutionists are required to assume continuity. They have to. It's part of their whole scheme, or the whole tree of life thing falls apart if they have discontinuity, which is why they almost never talk about discontinuity. Creationists believe in discontinuity, and I agree, there is discontinuity, and I expect there will be discontinuity between created kinds. The problem is, we can't assume that just because discontinuity exists that it defines the kind boundary. Right? And I like the analogy that Paul Taylor uses to kind of illustrate, not this, but I've repurposed it. It's a logical fallacy, where he says, if if it's raining, then the streets are wet. That's an obvious statement. We know this, right? But let's just say that we start with the premise that the streets are wet, and deduce from that it is raining. Can we do that? No, we can't. You know why we can't do that? Things like this. Yeah. All you have to do is dump a water bottle on the street, or spray it down with a hose, or have a fire engine come by, or maybe a water main has broken. Like, there's any number of reasons the street could be wet. It's not the only reason. Rain is not the only reason. For the same reason that discontinuity, if there's discontinuity present, it does not necessarily define the edge of a baramin. Matter of fact, it may or may not. Right? And this is why I don't really like the discontinuity criterion that Wood and Wise and those folks use, because I don't think it's logically coherent. Moving on down the Wilson paper. Disagreements over conclusions drawn from hybridization studies, stratomorphic series, and statistical baraminology have led to an impasse. It's not that these studies do not show useful patterns and groupings. They do, but there is a difference of opinion in what these patterns tell us. Consequently, I think we must move to something else more objective. Bingo, thank you, I agree. Something more objective. Statistical baraminology is a dumpster fire. It is a living, breathing dumpster fire, and it should have been burned up and thrown away 30 years ago. But we're living with the consequences of nobody wanting to tell Kurt Wise no. Unfortunately, this is part of where we are. So here's moving on down the Wilson. Sorry, Dr. Wilson, I don't mean to insult you. But here's moving down the Wilson paper. I propose that differences in genetic information content is the best metric to use in ascertaining baramins. The reason for moving away from morphology, as mentioned before, is that differential gene expression can result in mildly to extremely different morphologies in genetically identical organisms. Bingo. Differential gene expression can do this. Okay? The problem here, and I'm going to illustrate this just a little bit, is, again, this is slightly divorced from Scripture. It's away from Scripture because he's not taking into account hybridization. I still think hybridization is the best way. But I also recognize it's not necessarily practical. So I like this. I like this thinking outside the box. Let's get into the genetics. Let's use the genetics. Let's make something happen with it. Okay. Now, and here's his next sentence, or the next section I want to read. The goal of a baraminologist should be to distinguish, and this is the tricky part, between what genetic similarities are due to common descent and what is due to common design. Within one real baramin, the genetic similarity should be at or close to 100%, which also means that it is most likely due to common descent. Now, this is where I disagree. Because at or close to 100%, I don't think that's going to be possible. Because there's been a lot of mutations since the fall. A lot of them. And there's been genetic drift, there's created heterozygosity to deal with, and then genetic drift and mutations. So I don't think 100% is a reasonable number. I think 95% is probably pretty reasonable, though. You want to go with 95% or higher similarity? I think that's pretty reasonable. I mean, chimps and humans are much less than that. So 95%, probably fairly relatively reasonable. Now, of course, the evolutionists are going to say, well, chimps are 99%. No, they're not. That's based on a junk chimp genome and only comparing it to the portions of the human genome that actually matched up. They left out chunks of both genomes. The real number, based on actual research, is about 85%. So, the 99 or 95% similar, no. But 95% in the same baramin, at least with animals, this is reasonable. I think it's pretty reasonable, honestly. I think this could be adapted. I think he's thinking in the right direction. And maybe we pick a number. The p-value, accepted p-value in science is 5%. So maybe we pick 5% or less, or something like that. It's consistent. I think there is potential here. If nothing else, there's potential here. So here's where he spells out his criteria. We'll move down just the paper just a little bit. The genomic equivalence criteria. Because naturalistic processes cannot generate new genetic information, it would be good to consider this when looking at any taxon under consideration for baraminic status. Hybrid data or not, we must ascertain whether or not the genomes of all these species are essentially equivalent. What I mean is this: there needs to be close to 100% equivalence of both coding and non-coding DNA in the taxon being considered in order to tentatively conclude that they are within the same kind. I disagree with the 100% number. I think it's too high. Particularly because of gene loss. Gene loss can happen. It's not new information, but different organisms. Parasites are notorious for turning off genes. They can just throw them out. Because why? Because they're living a parasitic lifestyle. They don't need certain genes anymore for free living, so they don't need them. They just get rid of them because it's easier to replicate. So that's not a gain of information, that's a loss, but it's beneficial because it saves them energy. So we have to consider gene loss. This is why I don't think 100% equivalency is acceptable. I think we'd have to go with a lower number. I don't know what that number would be, but I think that 100% genomic equivalency just is not feasible because of gene loss, because of polyploidy, because of various other problems that could arise, mutations, genetic drift. I don't think we can get 100% equivalency. But again, 95 might be something to play with, and maybe we look at this and we say, okay, how do we do with gene equivalency? What's the equivalency look like, in particular species? Now, again, we may not have the data for this too, which may also be a problem. The standard genomic equivalence should be calibrated by the amount of difference exhibited in the human genome across all people groups, which is roughly 0.1%. All humans are about 99.9% the same. Why so strict? Because this is how creationists separate humans from the apes. It is because the mutation in natural selection cannot generate novel genetic information. It doesn't matter how small the increase is. If species 10 has more complex specified genetic information, the percentage is irrelevant. Whether it is coding or non-coding is irrelevant. Then species 1 to 9, they should not be placed together in the same kind. Again, I disagree with this, but I think he's thinking in the right way. Because I think that he's being too strict. He's not accounting for gene loss, but he's thinking about using genetics in baraminology, which I am all for. So I see where you're going, Dr. Wilson. I really do, and I like some of the stuff you're saying. I just can't go to that 100% gene loss as a problem. I can't. how do you deal with gene loss? How do you deal with the possibility of gene loss and degeneration since the fall? I think that's what he's ignoring here. And if he's ignoring that, that's going to make the model unworkable. But if we might be able to tweak this to get something out of it. Simple distance matrix. I mean, some programs will already calculate this for you. Eugene will calculate it. If I throw a bunch of gene sequences into Eugene, it'll calculate the distance for me. It'll calculate how far apart, how distant they are for those sequences after the alignment. So maybe a project for over Christmas break. Hint, hint. Anyways, down a little bit. He lays out a couple of possible complicating factors. One being karyotypical differences or similarities. If chromosome number differs, it does not necessarily indicate genomic non-equivalence. It may be due to a chromosomal fusion or other chromosomal mutations. In other words, you still have to look at the DNA in those instances. Again, information content's the issue, not necessarily where the information is packaged differently. Again, I would agree with that. Chromosome number is different in the dog kind. The foxes have wildly different chromosome numbers than some of the dogs do, but they're considered the same kind. Now, I have questions about whether foxes and dogs are in the same kind. But dogs are definitely in the same kind, and even they have and the fox, even all the foxes would definitely be the same kind, based just on morphology at least, and they have significantly different chromosome numbers. Endogenous retroviruses, ERVs. Genomic differences due to the number, type, and placement of retroviruses is not necessarily an indication of genomic non-equivalence. Conversely, similarities in the number, type, and placement of ERVs do not necessarily indicate equivalence. So basically, you need to actually examine each case individually in the case of ERVs. Gene duplication and allelic diversity. Differences in the number of copies of certain genes and or alleles do not necessarily indicate non-equivalence. Again, information content is the issue. So again, he's sticking to the information content thing here, which I like, but gene loss. Gene loss. That's the one thing he doesn't address here, because the next thing he talks about is ploidy difference. Gene loss, if you use 2 copies of a gene, that doesn't necessarily mean that you're not the same kind. Like, I can't... I can't go there. Ploidy differences. 2 species may have different ploidy, but may show clear genomic equivalence. And he talks about 2 species of frogs that illustrate this. Then he talks about the synapomorphy criterion from Kurt Wise. Wise, 1992, postulated the synapomorphy criterion, but with the advent of statistical baraminology, it has largely been overlooked. A synapomorphy can be defined as any uniquely designed trait, internal or external, that is indicative of and shared by all members of the same holobaramin. This is basically things that make a cat and make a dog, right? There are specific synapomorphies, specific criteria that are required, right? For example, he says, we should not assume Viperidae is a holobaramin unless we can show genetically that the vipers have the gene networks encoding them as the pit vipers do. So because the vipers have the pit organs, and the pit vipers have the pit organs, and the rest of the vipers do not. But they might still be the same kind, is what he's arguing here. See, what he's doing here is he's arguing for weighted traits, which, by the way, is something I would like to try. I would like very much to try using weighted characters, and I have tried it to a certain extent. Problem is, it takes a lot of weighing, so you really have to put your hand on the scale. I've played around with it a little bit. We may do some more work with it, though, and maybe at some point we will actually put weights on the scale, like he's talking about with synapomorphies. Moving on. Evolutionists say all life has one common ancestor, and creationists claim that God created the various kinds. If the evolutionists respond with the charge, where are the discontinuities, and we say there are indeed discontinuities, we just cannot agree on where they lie, and we cannot agree on how to determine them, then we have a problem that needs to be addressed. I would agree! This is why I'm working on addressing it. And he also points out the papers from Center in 2010, where he used statistical baraminology to prove that a bunch of dinosaurs that were clearly not the same kind were the same kind. So I just... Wilson did his homework for this. He knows about the Center papers. He knows that statistical baraminology is a dumpster fire and we need to fix it. Now, he's too nice to say it's a dumpster fire, so I will. It's a dumpster fire. One method in statistical baraminology is classical multidimensional scaling, which is CMDS or MDS. I just use MDS. One of its shortcomings is that of scale. Mace and Wood, using CMDS, showed that toothed whales clustered away from baleen whales in morphospace. If we select morphological datasets indicative of smaller taxa, is it possible to have dachshunds cluster away from Great Danes, and yet they are the same species? By the way, yes, it is. If we select larger taxa, is it possible to have chimps cluster with humans? Yes, it is. It seems that this is a real pitfall given that the Archaeopteryx clusters with early birds and coleosaurian dinosaurs using a certain dataset. Again, Wilson has hit on a really, really weak point of the model. Scale. And this applies not just to the MDS, it applies to the distance methods as well. Because they show distances, but distances will be viewed relative to the scale. So, right now, across the road from me is a house. You can't see that house, but it's there. Okay. For me, that distance between me and that house is probably about 100 yards, maybe a little bit longer. It'd be a good run for me to get to that house. But if you viewed me and that house from space, that distance would collapse significantly. Again, problem of scale. Because you're further away, the distance looks much smaller. And so if you're viewing a big group, distances will shrink. Yeah, it might explain some of the results that some of the statistical baraminology on dinosaurs got. So again, it's important to understand you can make these methods say anything you want if you pick the right data set. And it's been done, and I know it's been done. And when that paper comes out, we're going to walk through it. It's going to be a long walkthrough. It'll probably take more than one part, but we're going to walk through that paper because it has done an excellent job of exposing how bad some of this statistical baraminology garbage is. A couple more quotes. We need a criterion that distinguishes between larger taxa above kind, smaller groupings beneath kinds, and kinds. I propose that genomic equivalence is the answer, if we calibrate it by the taxon we are most sure is monophyletic, humans, i.e. modern humans plus all members of genus Homo, that knowledgeable creationists agree are descendants of Adam and Eve. Again, knowledgeable creationists might mean including Australopithecus sediba. No, sediba is not human. And Naledi's probably not human, and Florentius probably isn't human either. Erectus probably is, and Habilis is waistbin taxa. So, who knows what Habilis is? Just another quote here. Genomic equivalence with corroboration by the synapomorphy criterion can make distinctions that I believe statistical baraminology cannot make on its own. Genomic equivalence should always be informed by the natural way we distinguish between forms of life, which is by Gesalt, formalized as the cognitum concept by Sanders and Wise in 2003, which, by the way, the cognitum concept is a valid concept. I think it's one of the few things that I agree with Wise and Sanders on. This concept is a useful concept because it allows you and me, the average person, to sit down and go, yeah, those look pretty similar. They're probably the same kind, right? And he goes on, cognita are useful because of their correlation with synapomorphy. Synapomorphies are the objective, novel distinctions that often give cognita their recognizability. In other words, they're the traits that make a dog, and a cat, and make them stand out to you. If you look at it and go, that's a dog, that's a cat, that's a armadillo, right? You can pick them out. Why then is genomic equivalence informed by Gesalt and supported by synapomorphy superior statistical baraminology? Here's why. Here's his argument. They are derived from methods that we use to separate ape from man from those animals most similar to him in form and genetics, the apes. There is no doubt that statistical baraminology reveals real patterns and distinctions between groups and therefore can also, like Gesalt, inform the genomic equivalence criterion. However, I think that reliance on morphological data alone prevents the researcher from really knowing whether or not those discontinuities are 1. Between holobaramins, the ones we're trying to ascertain. 2. Between higher-level taxa, e.g. phyla, classes, orders, etc. Or 3. Between lower-level taxa, e.g. between species in the same genus or subspecies within the same species. It just may be true that hybrids can form between species that are apparently non-genomically equivalent. I would disagree with him here. I think that potentially, because if he goes to this genomic equivalence as the equivalent of a baramin. If he doesn't, if you don't go there, then it's possible I wouldn't have a problem with it. So, very interesting paper from Wilson. I am hopeful. I like the direction this paper's going. I very much like it. As a matter of fact, I'm probably going to send Dr. Wilson an email and see if I can work with him a little bit, because I really like the way he's going. I don't agree with him 100%. I think that he's going to end up separating species that are closely related because of the stricture which he's imposing on the model, but I like where he's headed, and I like what's going on. Stay with this. We're going to talk more about genomic equivalence and baraminology because I really like what he's doing, and we're going to mess around with it in Eugene and Phylocode and some of the other Mega and phylogenetic editing software and see what we can come up with. All right, thanks for tuning in, folks. Happy New Year. This is going to go up close to the New Year, so Happy New Year, and I will see you all in the next one.